A scientist who studied cosmic rays beyond the solar system also led more than 1,700 weekly birdwatching walks on his university campus. Alan C. Cummings has been a senior research scientist at Caltech since 1973. He worked on Voyager's cosmic ray subsystem, studying galactic cosmic rays in interstellar space. Cosmic rays are high-energy particles arriving from space. In 1986, Cummings and Ernie Franzgrote co-founded Caltech's weekly birdwatching group. Cummings had already watched birds for nearly 20 years. Members met weekly, observing and documenting birds across campus. By 2023, the group had completed more than 1,700 walks. That is a repeated observation record spanning decades, rather than a single survey. Cummings meticulously recorded every sighting, allowing the campus data to track long-term bird population trends and seasonal patterns. Voyager measured cosmic rays in interstellar space. His bird record stayed close to home. The notes preserve changes that occasional sightings would miss. A normal forest in northern Wisconsin became a 1,440-acre nuclear radiation research site in the 1970s, then returned to public forest when the experiment ended. Researchers wanted evidence for a practical question: how might a whole forest ecosystem respond after a nuclear disaster? They fenced the land near Enterprise in Oneida County and measured its plants, animals, weather, and tree growth. At the center, a cesium-137 source emitted gamma radiation. Beginning May 3, 1972, it irradiated the forest 20 hours each day, leaving 4 hours for research. The planned study was supposed to last 20 years, but budget cuts stopped irradiation after one growing season on October 16, 1972. Researchers still collected data until the project ended in 1974. The closest zone was not lifeless. Several lichens and some vascular plants survived. Lichens proved about 250 times more radiation-resistant than coniferous forests, a result researchers had not predicted. Eocarcharia was named as one dinosaur, but its fossils may represent at least two unrelated theropods, a spinosaurid and a carcharodontosaurid. The collection was not a skeleton. It was a group of isolated skull bones collected in 2000 from the early Cretaceous Elraz Formation in Niger. They came from separate skull elements with no complete skeleton connecting them. They were described in 2008 as one dinosaur. The name-bearing bone is a postorbital, positioned behind the eye. Later comparisons found that its shape and other skull roof bones fit a spinosaurid rather than the original assignment. The maxilla, a main upper jaw bone, and its teeth tell a different story. They belong to a carcharodontosaurid, a shark-toothed theropod group, possibly a distinct unnamed taxon. So the published name remains attached to a mixed fossil collection, while anatomical comparisons separate bones from different animals. The case is a reminder that a name can outlast its original arrangement. Linguists reconstructed two sounds in an ancestral Siouan language that was never recorded, nicknaming them Funny W and Funny R. That ancestor is called a proto-language, a proposed common source for related modern languages. Their forms become the comparison set even though no speaker or text survives from the ancestor. The comparison works because those languages share ancestry. Linguists compare cognates, related words descended from one source across those languages. Repeated patterns support a reconstruction. Natural changes, such as voicing between vowels or final vowel loss, help test whether that pattern is plausible. A reconstructed form gets an asterisk, marking it as inferred rather than directly attested. That symbol keeps an evidence-based proposal separate from a recorded word. So Funny W and Funny R are not recovered recordings. They model an earlier sound system through regular correspondences among related language descendants. By 2023, only about 150 people had been diagnosed with Scraben-Deardorff syndrome, yet one WDR26 variant can disturb nervous system development. The syndrome combines developmental delay, intellectual disability, seizures, feeding difficulties, and neurological and facial findings. Because these features overlap with other genetic conditions, appearance alone cannot establish the diagnosis. WDR26 helps assemble the CTLH-E3 ubiquitin ligase complex, a cellular machine that marks selected proteins for degradation. It also supports gene regulation by helping this complex reach DNA, so reduced functional WDR26 changes protein and gene activity. During nervous system development, that imbalance can disrupt coordinated cellular programs, producing the syndrome's recognizable developmental and neurological presentation. A single pathogenic copy can be sufficient because the remaining copy does not provide enough functional protein. Genetic testing confirms a pathogenic or likely pathogenic WDR26 variant alongside the clinical pattern. That result separates this syndrome from overlapping diagnoses and helps clinicians plan individualized, neurological, and supportive care. A plant genus known only from roughly 50-million-year-old fossils carries the name of Oregon's 1972 Biology Teacher of the Year. Bolenia is an extinct flowering plant in the soapberry family, known from Eocene sediments across western North America. Fossils come from the Okanagan Highlands and Colorado's Florissant Formation. Researchers reassessed fossil leaves, fruits, and leaf venation, the vein patterns inside leaves. Fruit structure mattered. Some fossils showed fruits borne in threes, unlike modern Dipteronia's typical pair. That combination separated Bolenia from earlier classifications. In 1987, researchers established Bolenia as a genus with two described species, Bolenia americana and Bolenia insignis. Later work kept it in the soapberry family while leaving its precise subgroup uncertain. The name preserves two kinds of evidence: a reconstruction of an ancient plant. It also links that fossil record to an Oregon biology teacher honored in 1972. Both now sit in one word, Bolenia. A biologist's home in Jena still contains lamps and painted ceiling ornaments inspired by jellyfish forms. The house is Ernst Haeckel's Villa Medusa in Jena, built as his residence in 1882 and 1883. Haeckel also used the villa as his workplace. Haeckel was a marine biologist known for richly illustrated presentations of marine life. His books rendered organisms as carefully observed, artistic forms, making jellyfish structures available as visual motifs inside the villa. The dining room makes that connection concrete. A painted ceiling decoration uses Haeckel's own drawing of a jellyfish found in Sri Lankan waters. It is a scientific image adapted into architecture. Some lamps and ornaments have survived. In 1920, the residence became an archive and museum, preserving Haeckel's papers alongside these interiors. The archive contains around 38,000 letters and other documents. Today, it houses the University of Jena's Chair for the History and Philosophy of Science. Guided tour let visitors encounter scientific drawings as part of the building itself. A former mail carrier sold a plastic dumbbell as a disease cure powered by a force unknown to science. Those claims ended in a fraud conviction. In the 1940s, Ferguson promoted the Zerit applicator through mail order and a network of salespeople. He advertised it as an atomic cure for arthritis and rheumatism. The blue and white device looked like a small dumbbell with two plastic globes at its ends. Inside were paraffin wax, dust, and tap water, not a demonstrated way to heal anyone. After a customer complained of no result, police arrested Ferguson and associates in 1948. A federal case followed over misleading interstate medical sales, while a post office fraud order barred mailings for the device. In 1950, Ferguson was convicted of fraud in court after a Geiger counter found no radioactive material. Chemical analysis matched Chicago tap water while doctors found no therapeutic value.